Hello and welcome everyone. Thank you for joining us for today's webinar. I would like to thank all my viewers for making our webinar series a great success. This reminds me actually of one saying individually we each are one color, but together we are a beautiful rainbow. Myself, Dr. Sakshi Bhadraj, application scientist, Eltem Technologies. Today we are presenting a sneak peek into the future with our today's masterclass on redefining junk, the Junkomic approach of computational drug design. Yes, you all heard it right actually, the junk. We all know about junk food, right? So what is junk in drug designing? Do we ever think about the junk portion of a genome? Do you ever think to explore the potential of these unused junk regions to design the life saving vaccines and drugs using the computational approaches? Could you believe that a small portion of human genome that is likely to be functional but does not code for the proteins that is a junk? And this junk is now an attractive hotspot in the research of novel functional molecules. So, to understand this junk and gencomics better, we have a specialist with us today. Our guest speaker for this masterclass will be talking about the gencomic approach of computational drug designing. So stay tuned during the session to learn something about these gencomics and how we can utilize them for the drug designing. Right, feel free to drop your questions on the topic. We will take up all the questions at the end of the session. So, without any further ado, let me introduce you to our distinguished speaker of the day, Dr. Navya Raj. Ma'am, working as Assistant Professor, Department of Health Informatics, College of Health Science, Saudi Electronic University, Dammam, Saudi Arabia. Ma'am has 15 years of exposure to the bioinformatics and computer-aided drug designing. She also worked as bioinformatic research analyst with Science Bioscience Research Institute, Chennai. She has completed her MPhil and a PhD in the computational biology and bioinformatics from the University of Kerala. She has expert hands in disease-specific drug designing strategies like cheminformatics, drug repurposing, structural bioinformatics, immunoinformatics strategies for the peptide vaccine designing, antigen antibody docking, amyloid beta based therapeutics strategies for the Alzheimer's disease as well. I welcome you, ma'am. We are so glad to have you with us today to share your insights on the redefining junk, that is, Junkomic approach to the computational drug designing. Over to you, ma'am.
So, good morning, all of you. First of all, thank you so much for this opportunity to interact with you all. So, I would like to thank Altum Technologies, Dassault Systems, especially Dr. Sakshi and Ms. Divya for this opportunity. So, I'm happy to be here. And yeah, let us start with the presentation. So this is the area which I'm working for the past couple of years. This was my area during my MPhil and PhD, and then I'm taking forward this area, working through for some new exciting results in this area. So as announced by Dr. Sakshi, the topic for today's session is the redefining junk, the Junkomics approach to computational drug design. So let me go to the next slide. So today I'll be giving an overview of what this research area is and what were the objectives of my study, the methodology we have adopted for this study, and what results we obtained, and I'll be then concluding the work. And after that, the session will be open for discussions and question and answers. So next slide, please. So we all know we are all in a world which is filled with information, we are working on a lot of information in different kind, and we the biologists who are into biology are having a lot of information inside the organism or inside the cell. So the information is flowing in biology in the form of different biomolecules, and we all know the key players in this information flow are the DNA, RNA, and protein, whereby we know the important concept, the central dogma, where the DNA, which is the life or the blueprint of life, that is inside the DNA, inside the cell, in the chromosome, and that DNA translates into protein. In between, there is a mRNA stage or RNA, and then it gives us the protein. So this steps like the DNA to RNA and RNA to protein is the central dogma theory, and it was first proposed in 1958 by Francis Crick, who was the discoverer of structure of DNA. So these biomolecules are the key players in the research, in any biological research. So next slide, please. So when we think about this particular era, we are in an age of omics research, because everything we want in a large scale, we are looking into everything, every biomolecule in a larger scale. So we aim to understand not a single biomolecule but as a network. Before, it was the approach we are learning about one gene or one protein, but now it is all networks. So we aim to understand everything as a network, and that is the omics approach. So we have the collective technologies to explore the relationship between molecules, and we know the recent technological advancements in all the fields have given exponential growth in biological data or information. So we need computational methods or approaches to analyze and manage this high throughput data. And for each stage in this information flow, we have given names for each stages. So people who are working with DNA or genes, they are into genomics, the omics of genes. And who are into mRNA or the transcripts, they are into transcriptomics. Who are working with proteins, they call it proteomics. And then we have the different cell functions, we have systems biology, you have many areas coming in that. But still, importantly, as we saw, genomics, transcriptomics, proteomics are the highlight areas in this omics approach. Next slide, please. So when we talk about a genome, it contains the coding region and the non-coding region. So the non-coding region are likely or apparently called as the junk region, which are not useful or which are not coding for a functional protein. That was the concept. Earlier concept, but now we know there are many projects came up, like the ENCODE project, which gave the idea that there are a lot of more number of transcripts which are actually giving rise to some proteins or peptides. And that is like again one more area, hidden area, which is the long non-coding RNAs, which is giving rise to the peptides. So some information is already there, some information we still don't know. So the non-coding regions are not like idle regions. They are doing some influence in the gene expression. They are having some switches to control these gene expressions. So non-coding regions and coding regions and the junk regions. So our lab, or we were interested in learning about this junk region. So what can we call these junk regions? Next slide, please. So these junk regions, when we are studying the junk regions, what should be the term given? So Junkomics is the right pick, right? We can call the study of the junk regions as Junkomics. Next slide, please. So redefining junk. It all started with work in Riken lab, Japan, where my mentor, my professor, Dr. Pavan Kethar, he's a professor right now in Jawaharlal Nehru University, JNU. So his colleagues and his team did a work, a redefining work for the junk, where the so-called junk was proved not to be a junk anymore. So 6 proteins were artificially expressed from the junk regions of the sequences. They did some wet lab work and expressed those proteins, and that proteins were studied and they were named as eka. Eka means first in Sanskrit. So that name was given for these particular proteins, and this was published in this journal of biological engineering in 2009. Next slide, please. So with that, from that work onwards, our lab started. So I was a part of his team way back in 2012, 11, 12 to 2017 in Kerala University. Computational biology and bioinformatics department. So our team carried out more extensive work on the junk sequences, and the Junkomics approach was further carried forward. So as we distinguish the data or the regions as the coding and non-coding, we can identify one more region, which is note coding regions. It means the coding regions gives to the mRNA and gives to the functional proteins, proteome, the total proteins. That is well studied. And you have the coming up region where the functional non-coding RNAs or regions are there, which gives some peptides or proteins which are having some functions. So that is coming up. Identified regions, or there is information, but there may be so many regions which is not having any history of coding any protein or peptide till now. So those sequences are available. So we can computationally do a prediction, a translation, a transcription of those sequences, or we are translating that into proteins, and the peptidome, the entire peptides from that region is being studied. So you can find out what are the functions of it. Still, if some peptides are available which no known functions, no similar functions, so we can use these sequences for some possible applications. That was the Junkomics approach. Next slide, please. So now, once we computationally work with these junk sequences, and we get a lot of peptide molecules which are stable, which are novel, and we are making a database of these peptides, new molecules, but without any known function or any history of translation, anything. So that these peptides we are getting as new, fresh molecules. So what can we do with this? So we thought about, why can't we employ these peptides in the drug design aspect? Because peptide drugs are very much into a market, or they are entering market. Many are into clinical trials, and it is a good plausible alternative to the small molecules as well. So that was the thought which initiated in this study. Next slide, please. So the key objective of this particular work is to computationally predict and analyze the putative peptidome, the possible peptidome, the entire peptides from the intergenic regions of a bacterial genome. So any junk regions we specifically selected, intergenic, the regions between one gene and another gene. There are 2 genes, and you have in between one intergenic non-coding region. Those regions we have selected. And from that, we generated the entire set of micropeptides, small peptides, which are not much studied or very less studied. So the length is less than 30 amino acid. And those peptides are known as micropeptides. And then the structural and functional annotation of these novel peptide molecules were carried out computationally. Then to use it in a drug designing perspective, the drug likeness prediction was done, screening was done based on the stability of the peptides and toxicity of the peptides, because for any peptides, the stability and its toxicity is the major setbacks when it goes into the clinical trials or in the further stages. So we tried to predict it online, and applications of these novel peptides as candidates for drug designing perspective, drug designing studies were carried out for a particular case study, selecting some particular protein targets. Next slide, please. So we selected the model organism, basic model organism, E. coli, Escherichia coli, K12 genome, and then you all know about E. coli. It is a rod-shaped gram-negative bacteria, circular genome it is having, and the 4.6 million base pairs it is having in the genome. And annotated protein coding genes are 4,466, and 12% of this genome is non-coding DNA. So that 12% intergenic DNA, you can see the intergene there. One gene, a functional gene, another gene, the between portions is the intergenic region. So that regions we focus, those sequences we focus for the study. Next slide, please. So this was the methodology adopted. We took the intergenic sequences, downloaded the intergenic sequences set. It was about 2,500 sequences. Then we conducted a BLAST search, which is sequence similarity search against the non-redundant sequence database. BLAST X, it will do the translation of the sequences and then do the BLAST. So those sequences which doesn't have any hits, no hits, without any similarity with the existing database, we selected those sequences and we did a 6-frame translation using the emboss translate tool, and then selected the neat frames, the best frames, which length 5 amino acids to 30 amino acids, the short frames without any stop codons in that. And then those short peptides were again a BLAST, conducted a short peptide specific BLAST to see whether any similarity is there in the database. So we adjusted the word count and the e-value as per the small peptide BLAST. And then from that, whichever sequences were not having any similarity, no hits, those sequences alone we further we took for the physicochemical study, like the physical aspects and chemical aspects of it, using the PROT-PARAM tool. And from the in that again a sequence-based similar stability will be predicted, and those peptides which have good stability in the sequence level were carried forward for the drug likeness prediction. And drug likeness we predicted using a toxicity prediction, ToxinPred tool, a server. And from that, again, those peptides were giving good results. We carried forward for an intestinal stability prediction, the half-life prediction server, HLP server. And in that, again, the stable and non-toxic peptides, highly stable and which are non-toxic, we modeled using INITIO. Because we don't have any similarity, we modeled it in an ab initio approach. And then we studied the Ramachandran plot, stability of these peptides in the Ramachandran plot, and then those peptides were added into our in-house database known as ECA knowledge base. Okay, next slide, please. So coming to the results part, we have taken around 2,500 sequences. Out of that, 1,860 sequences were having some similarity, already some similarity in the BLAST search. And you can see the numbers here. Some sequences were having similarity with the known proteins, already the known proteins, and there were some similarity in hypothetical proteins. Some were known hypothetical from other species. So there were that was classified and that was kept aside because they already have similarity, so we don't need it. We need those sequences without any hits. So we had 649 sequences without any hit in the BLAST. So those sequences were carried forward. Next slide, please. So those sequences were specifically done for the short peptide BLAST after the translation. So we got 4,900 neat frames. So that around 600 sequences we translated into 6 frames. We got 3,894 sequences. From that, we took only the best frames. So you can see when we did the BLAST, short peptide specific, 1637 sequences again had hits in short peptide BLAST-P. So those were again cataloged with, most of them were from the known protein and non hypothetical proteins from other species. So that also, those sequences are also kept aside. We didn't use those. We used the other sequences which didn't have any similarity. Next slide, please. So from the BLAST P short peptide, without hits, 3263 sequence without hits in the short BLAST P, we carried forward through the param prediction, and a biopearl module was used there, and 1558 stable sequences were obtained. So those out of these 3000, we got around 1705 sequences were predicted to be unstable. So we again we didn't take it forward. We took the 1558 stable micropeptide. So in the proparam, the molecular weight, everything will be predicted. Along with that, we will get a instability index. So if the instability index is above 40, it is unstable. Below 40, it is stable in vitro. So that condition we have taken. That is a default one, and that we have taken. So the molecular weight of the sequences were found in range 400 Dalton to 3500 Dalton, and all these 1558 stable micropeptides were added to our ECA knowledge base, and they were named as ECA micropeptides. Next slide, please. So the intestinal stability prediction carried out through the HLP server, and here the stability was about 66 peptides, and they give stable based on it will predict high stability or low stability. So the highly stable peptides were taken, and you can also see the half-life, so which is having a good half-life, which will be like available for as peptide for bioavailability with a good half-life. Those peptides were carried forward. In the next step, next slide, please. So toxin pred, which predicts the toxicity. So toxicity prediction, out of 66, around 65 non-toxin peptides was predicted as non-toxin. One was predicted as toxic. So the other 30 out of this 65, then we saw with the basis of the length, because for modeling the peptides, we need at least 10 amino acids initially. So we started those peptides, and from the results of the high stability in the HLP server and the toxin pred, finally we selected 36 sequences to be the best one to be modeled. And some we modeled, we didn't get good structure, so that also we avoided, and finally non-toxic and highly stable 36 structures sequences were modeled using ITSER. Next slide, please. So here the RC plot or Ramachandran plot was calculated for these peptides, all the peptides. We saw how many amino acids in the core region, how many are in the allowed region, how many are in the disallowed region. So based on that, again, we sorted out which are the best peptides. Next slide, please. So finally, these are the structures, the ab initio predicted structures of the 36 EMPs, ECA micro peptides predicted using the Junkomics approach, and it was added. So it had all the properties also, how much is the molecular weight, how much is the half-life, how much is whether it is toxic, whether it is not toxic, all the details we have. Okay, next slide, please. So by the time there were studies coming up about micropeptides. So what are actually micropeptides is they are short, unprocessed, non-classical functional peptide products of the short ORFs, open reading frames. Okay, and preferably they mostly are coming from the non-coding portion of the DNA. And so initially they were thought to come from the long non-coding RNAs, but now those regions are known as the transcript of the unknown function. They don't have any function, the known function. We don't know the function yet. They may have some function, but right now they are not known. and they mostly are filtered out during the genome annotation methods. When they are done, these short peptides are normally discarded, but we have taken those peptides and we have studied the possibility of using it into a next level. So these peptides normally don't have an N-terminal signal, and they are immediately released into the cytoplasm, and the length is about 2 to 30 amino acids. And the first eukaryotic micropeptide which was identified is known as the early nodulin 40, E-node 40. It was identified in soya bean plant. So there were studies coming up, and there were reports that these micropeptides are the next best thing after microRNAs. So it has got function, some of which are being identified. They are having some functional importance. So which don't have any importance, we have studied for a next level. So next slide, please. So we conducted a case study for those peptides which doesn't have any similarity with any known peptides and doesn't have any known function right now. We applied these micropeptides as drug candidates against Alzheimer's disease. Next slide. So we selected Alzheimer's disease because of its global burden and so many cases we know, and there is no current any important drug for Alzheimer's coming up. There are many drugs in the line in the clinical studies, and some of them are in the market, but still a lot of research is going on, and we all know how it happens. There is the amyloid beta peptides formed, and that peptides become forms tangles in the brain cells, and thereby causing Alzheimer's disease. And the beta secretase 1 protein is a highly important target in the pathogenesis of AD. So that that protein target was selected for our study. Next slide, please. So the methodology here for this case study was we selected the target structure, we downloaded it. Many structures were there. We downloaded the important ones, selected ones with a non-peptide inhibitor so that we can have a comparison. And the binding site was identified using Discovery Studio. Then enzyme inhibitor docking was carried out using ZDock module in Discovery Studio, and also an online tool like PatchDock. And the best interacting micropeptides were selected, and the best one, molecular dynamic simulation, was also carried out. Next slide, please. So this is the superimposition of the different base 1 structures, the beta secretase structures. 3 structures like I1FK, N2 ZHR, and 1X N2. 3 PDB structures were downloaded, and those structures were superimposed using Discovery Studio. And you can see the center portion where the binding site is. So we can identify the important binding residues there, and can see where these the ligands are normally bound to these particular target structure. So for identifying the binding site, we used this step. Next slide, please. And then we carried out the docking using the ZDock tool and the PatchDock tool. So from the PatchDock tool, we will get a global energy value, and the ZDock we get a ZDock score, as well as the energy based on the energy based function. It will do a ranking also, a Z rank, which will tell you based on energy which is the best docked molecule, the pose. So based on all the 36 different micropeptides, we selected the best 5 based on the global energy, ZDock score, and the Z rank score, and the number of interactions. And these were compared with the native peptide OM99, which was the first peptidomimic peptide inhibitor identified for base 1. So that was taken for the comparison, and we redocked that also, and with that values we compared with which are giving better scores, and those 5 peptides were shortlisted. So this is the structure we obtained after docking, the complex we obtained from the Discovery Studio, ZDock. So the green color molecule is the EMP5, which is our micropeptide, and it is how it is docking inside the base 1 cavity. Next slide, please. So you can identify the binding residues also in Discovery Studio. You can have a lot of graphical representations, and you can analyze the molecule in a very good quality with the residues, which residues are interacting with the peptide in the binding site, how it is properly interacting, what are the atoms interacting, the bonds formed between these peptides and these amino acids that can be analyzed. So that we have analyzed. Next slide, please. Next slide, please. So this is a table where the molecular interactions of the best peptide and the receptor is being identified. So from the Discovery Studio, you can get all the details of what amino acid is interacting, what is the bond formed, what type of bond is formed, and what is the distance of the bond in Armstrong, like how much is the distance between the distance of this hydrogen bonds and all. So you can see which is the best bond, which is the shortest bond, that is good, that is having a good contact. So and even the non bonded contacts or hydrophobic interactions, everything can be analyzed. So we analyze for the best peptide. Next slide, please. So then a short step of standard dynamics cascade using the Discovery Studio was performed for molecular simulation run. This is just for 10 NS, but we are doing the a longer run with 100 NS we are currently doing. So this was an initial study with just 10 NS, and we got a good result for that also. So this is a standard dynamics cascade where it will provide the different steps of the equilibration and for all the for carrying out a molecular dynamic simulation. So next slide, please. So you can see the energy, total energy, and it is keeping constant, or it is not much fluctuating for the 10 NS time. But now we are moving forward with the 100 NS or more longer run to identify whether it is stable. So from the initial result, it is good, showing a good stability with the micropeptide, and the target is showing a good stability. Next slide, please. So final deliverables of our study is a multi-parametric in silico library is constructed with 1558 novel micropeptide molecules with the molecular weight in the range 400 Dalton to 3500 Dalton, which is a good window, which is easy for chemical synthesis. So you can easily select these peptides which is having good interactions or good properties. You can chemically synthesize it and then use for drug designing purposes or drug development purposes. And out of this, 35 non-toxic stable micropeptides were predicted with high intestinal stability. And the best interacting micropeptide was identified as a potential lead molecule for base 1, which can be used in a drug development pathway for Alzheimer's disease in the future. Next slide, please. So in conclusion, this is was an in silico study, and it suggested that you can tap the hidden potential of the junk regions, or which are the less explored landscapes of genome, and you can have some therapeutic offerings from this junk. Junk can give you jewels, so it can give you some interesting therapeutically viable biomolecules which can be used in a synthetic biology perspective. And this workflow was adopted to show like how a piece of DNA which is considered as junk, and we were like keeping it aside, can be fine-tuned, designed, polished, and made into a potential therapeutic micropeptide, or like a drug molecule, like a small therapeutic molecule. And so further in vitro and in vivo experimentation is needed. We are in the process of it, and that needs to be done for identifying in the interactions in the molecular level, in the pharmacological aspects of it, and then use it in the synthetic peptide drug development. Next slide, please. So an initial data or the preliminary information is already been published, and this was in synthetic systems and synthetic biology journal. Two more publications are on the way. Already one is already been submitted, another is already been accepted. The abstract is accepted in a special issue. So we hope that we can come out with new publications in the near future. Next slide, please. So for all these activities, for all this project, as well as I was involved in some collaborative research also. Along with this particular Junkomics approach, we have also done some other structural bioinformatics studies, in which the Discovery Studio tool has helped me a lot. Actually, I started working with Discovery Studio from 2007 onwards, and I really love the tool. Like there are so many options in that, and I'm more interested in the structural aspects. So I work a lot with used to work a lot with this docking studies and peptide designing, all that. So these are some glimpse of my other collaborative research projects which we have carried out using Discovery Studio. So in this particular study, we have taken a molecule like radicicol. This was a collaborative study with the University of Hyderabad. And radicicol, which is a drug used for malaria, and that we modified. We made different, say, analogs of this molecule, and then we tried to dock it with the topoisomerase 6B, and this is a publication which came out of that collaborative work. Next slide, please. This is another collaborative work with in the University of Kerala, where we studied with plant-based compounds. This is of small molecules like camphanol, then imodin. So these compounds, we tried to study the interaction with the EGFR, which is for the cataract eye protection. For cataract, these compounds are highly important. So that is again a collaborative study used with Discovery Studio. Next slide, please. So this was yet another work done with Osmania University, where antifungal peptides were studied for their pathogenicity against Aspergillus flavus, and that there also the interaction. You can see that even in Discovery Studio, you can see give the membrane. You can give membrane related representations for the protein. So you can see the extracellular, intracellular regions, and the pink color, the magenta color is the drug or the synthetic peptide which is going inside the membrane, or it is half inside the membrane, half outside the membrane, and interacting with the giving the with the peptide with the target protein. So these are some of the studies which I have already carried out. Next slide, please. So basically, I basically like to work with protein molecules, structural bioinformatics. So from the sequence level, we can come into, say, the colorful world of proteins, peptides, and interactions. So that is the important area which attracts me a lot. So Discovery Studio has been a very important part of my research for a long period of time, and I really enjoyed working with it, with all the colorful molecules. So you can enjoy the colorful world of biomolecules when you are into this area of research. So with that note, next slide. I'm concluding where this talk was just a glimpse of the work, what my interesting area, and how say the Discovery Studio has helped me to generate these wonderful results. So junk regions, we can be focused as important resources, as a template for finding out new peptides or new molecules and their interactions. So as Dr. Sakshi told, junk food is not good, but junk DNA will always be good. So thank you for listening. So I hope it is clear, and you got a little bit of enlightenment in what this area of research is. And thank you so much.
Session. I hope all my viewers enjoyed this amazing session. Now it's time to take some questions. So we have our first question. Which interaction is more significant? Hydrophobic, alkyl interaction?
Actually, it depends. Is it audible? Yeah, hydrophobic interactions are good, and alkyl interactions are more. We have to see with the distance. Say how much is the distance between these molecules. As I showed in the table, you can see the how much close vicinity these are, and how much strong these interactions are. Based on that, you can select which are the best interactions. So both alkyl and pi interactions are good. I think alkyl interactions are more favorable ones, more commonly seen ones, I think, in the hydrophobic interactions. Is any significant of taking small chains of peptide 5 to 30? Yeah, that's a good question. Actually, this range 5 to 30 peptides are very less studied. So we had a previous work where we have taken the peptides more than 30, and that is the normal window which we take for peptides. Like more than 30 means 100 base pairs, more than 100 base pairs. So almost all the gene prediction tools and every work focuses on those area of proteins and peptides which is more than 100 base pairs, so more than 30 amino acid, like 40, 50, that is the normal range. So that particular study we have already done, and then this particular study was focusing on the lower range, which is less explored region. Is the kind of protein docking. Yeah, it is peptide protein docking. Peptide and peptide you can dock, or peptide and protein you can dock. This is a protein docking. Any more questions? Am I audible? Is the connection on? Hello, Dr. Sakshi, are you there? Is there any more questions?